pleasure today of introducing our student-invited speaker for this semester, Dr. Bree Rosenblum. Bree is Assistant Professor of Environmental Science Policy and Management at the University of California, Berkeley. And I really think it's the breadth of research topics in her lab that made her such an ideal choice to appeal to the diversity of interests among the graduate student community here in EEB. Bree received her PhD from UC Berkeley. She completed her postdoctoral training at UC Berkeley, and after taking a job as assistant professor of biology at the University of Idaho for a few years, moved back to Berkeley where she joined the faculty in her current department. It's clear that Bree is deeply committed to mentoring young scientists and to her teaching, both of which she's been recognized for at the university level multiple times. And it sounds like her success in these areas could largely have been influenced by her early experience as a middle school teacher. She's also been a yoga instructor for many years and reportedly does a mean version of the worm, as in the breakdance move, which we may or may not get a taste of at a happy hour dance-off immediately following. So, needless to say, we are very grateful to have the opportunity to hear some of the latest research from her lab. So please join me in extending a warm welcome to Dr. Bree Rosenblum.
Hi, thanks everyone for coming today. Is the mic on and okay? Great. So I like to start out just with a brief blank slide acknowledgement just to thank everyone for getting here and taking a moment to thank any modes of transportation, people that facilitated, stood in your way, but we're all here. And also to thank the people that brought me out. It's been really lovely to have John and Clara as hosts, and Tommy's done a lot, and loved getting to interact with the grad students today and looking forward to more time together tomorrow. And also looking forward to kicking everyone's butt at the dance-off. So if you're an evolutionary biologist, you can't just kind of start with like, oh, good, thanks, I'm glad we all got here yesterday and our planes arrived on time. You kind of have to go back to like, I'm glad we all got here. And so I like to just start with a brief motivation. Most of us that are biologists have in some way an underlying fascination with how some part of the universe works. And I like to just give a very brief, which obviously is unnecessary for this audience because you all know how we got here, but a little run through of the history of life on our planet just as a way to motivate what I care about and what the kind of research is that I do. So our planet formed about 4.5 billion years ago. Things were a little dicey at first, but eventually we had what Dawkins calls the replicator with a capital R, like in Winnie the Pooh. You know how, like, the replicator. And once there was the ability for replication, life on our planet followed fairly shortly after. Life not only was influenced by the environment on the early planet, but also influenced the environment on the early planet, and went along until we start getting to the branches that we recognize: eukaryotic life, multicellular life, explosion of multicellular animal life. And then we come to some of the relative newcomers that many of us study: land plants, mammals, our own ancestors. And if you kind of scroll through time into, is there a little feedback? No, we're fine, just in my own head. We scroll through time, we get to our own personal histories and how those relate into the story of the universe. These are my great-grandparents before they came to America. And if we scroll far enough, we get to our own lives. This was how many people had a tuxedo shirt? No, yes, yes! I love this tuxedo shirt. I had this, it was like my band concert outfit, and I was so into my tuxedo shirt. And probably for most of us, you can identify, if not a moment in time, at least a period of time in your life when you realize not just that you could be amazed by the things around you, but you could actually study them as a profession. And so I like to think about this in my own life as life gets interested in life. You're like, whoa, I could actually do this as a job. And for me, this is a picture of me and David Rand's molecular fly genetics lab back in 1992 or something. And from there, some of us go on to have careers, but for me, the fundamental motivation in terms of really being interested in life itself is really at the heart of what my lab does. It's really at the heart of what motivates me as a scientist. And the way that my lab tends to study the diversity of life is really by an interest in the fundamental mechanisms that shape the tree of life. So my lab is really divided into two halves. Those people, it's like a wrestling competition, those people that are primarily focused on diversification, and we work mostly at recent timescales, but there's certainly been billions of years, and this is a cartoon tree that doesn't bear a whole lot of resemblance to what actually happened, but billions of years of diversification on our planet, which of course is balanced by billions of years of extinction. And so what I want to do today is give you a couple of research vignettes from the work that I do on these two themes. And I tend to flip a coin. I tend to flip a coin about which one I present first, whether like I want to start depressing or end depressing. And I'm not sure what I gauge about the audience to make that decision. But I think this time we're ending depressing, which is nothing wrong with that. So I'll talk a little bit first about a project that I've been working on since graduate school on adaptation and speciation, and also talk about a project that I've been working on since my postdoc about amphibian declines. And these two research themes are really tied by being both sides of this evolutionary coin of what facilitates the explosion of diversity on our planet, as well as what factors might prune diversity from the tree of life. So we're starting not depressing on the adaptation and speciation side. And most of my research takes these kind of fundamental evolutionary processes but puts them in a fairly modern context. So I don't really do deep time work. I'm mostly interested in how organisms respond to rapidly changing environments. And they're not all of my projects on anthropogenic timescales, but they all usually facilitate, they're all looking at processes that have occurred over the last few thousand years. And we know that adaptation and, in fact, even speciation can happen very quickly on our planet. We know that from artificial selection studies. We know that from human experiments with domestication. We know that because of how organisms are responding to anthropogenic disturbance. We also know that from examples of rapid evolution in response to recent geological changes. And as a graduate student, I listened to one of my mentors who said, come up with your big scientific question and then find a system that's really going to be great for answering it. And I remember spending the first summer of grad school just like driving around the US aimlessly, like I'm supposed to find a study system somewhere out here. And then I got to White Sands and I was like, oh yeah, I'm going to stay here for a while. Has anyone been to White Sands? Few of you. It's a really cool place. The lighting doesn't exactly do it justice, but these gypsum sand dunes are as white as a piece of bleached white paper. Gypsum's what they make drywall out of. This is really, really light substrate. And the gypsum sand dunes at White Sands, this is just an aerial photograph. It was fascinating to me because they're geologically very recent. Most of the gypsum has just been deposited in the last 2,000 years. And there's also no geographic barrier between the gypsum habitat, which looks like this, and the rest of the Chihuahuan Desert that looks like this. And so any adaptation that's happened in the gypsum sand dunes has happened despite the potential for the swamping effects of gene flow. There's no geographic barrier. Okay, and a lot of species exhibit local adaptation to these sand dune formations, and this was what really caught my eye, is that no one had studied. Since they had been described in the 40s, no one had gone back and studied this very unique lizard fauna at White Sands. So this is a picture of one of my favorite earless lizards, and this is a picture of it in its natural habitat. And I forgot, but I normally bring a lollipop for the first person that can find the earless lizard. It's small. I'm not really cheating, it's just small. Does anyone see it? You see it. So these guys are really, really, really well camouflaged in their environment. And of course, the thought is that these are small diurnal lizards. They are subject to intense visual predation. This is just a staged photograph of one of the species substrate matched, and this is one of the darker forms that I just put on the White Sands, just to demonstrate that if you are a small diurnal lizard that is eaten by loggerhead shrikes and other birds of prey that primarily are visual hunters, that it is a good thing to be camouflaged. And in fact, there's been all sorts of empirical tests that have supported this in terms of doing predation experiments and other things. So when I started the work at White Sands, I started focusing on the 3 lizard species, which are actually the only 3 lizard species that live at White Sands. There's dozens of other lizard species a half a mile away, but these are the only 3 that live at White Sands. There's all sorts of interesting ecology that I'm really not going to go into today. For example, there's density compensation, so that the number of total lizards at White Sands is basically the same as the number of total lizards down the road, even though there's only 3 species instead of 24. So there's a lot of interesting things happening at White Sands, but the thing that struck me, because I was interested in the possibility that you could have rapid adaptation and possibly speciation, even in the face of gene flow, if selection was strong enough, was that there was this replicated system where you had multiple species all evolving over very short time scales, this blanched coloration, compared with their ancestral forms throughout the rest of the Chihuahuan Desert. And these are the earless lizard, the fence lizard, and the whiptail lizard. And basically, the light and the dark lizards are different in every way that you can measure. And I'm not going to belabor this in a huge, lots of data way, but I'll just tell you that not only are they different in color, they're different in their morphology in terms of their body size, their limb length. There's differences in these morphology that actually translates into differences in performance that we've measured. So this is one of my favorite photographs. This is who's now an assistant professor, but was a graduate student with me, Simone, and one of her undergrad field assistants, Jack. And any herpetologists out there that know what's happening here? Yeah, come on. Jack is trying to motivate the little earless lizard to run, right? He's with a feather duster. And then they were videoing these, and then we can measure with high-speed videography how fast do the lizards run on different substrates. And we can see whether things like differences in limb length translate into differences in performance, like how fast can you run away when a fake bird is chasing you. And this I think is the only picture of all 4 legs off the ground for a little Holbrookia who is clearly motivated. There's also lots of differences between the habitats and resource use. White sands lizards are using microhabitats differently than their dark soil counterparts, even once you take into account differences in habitat availability. They differ in many aspects of their behavior, including things like wariness to predators, all in directions you would predict. They differ in circulating hormone levels, and the newest project that an undergrad just finished in my lab, which is so fun when someone comes to you and is like, I really like sectioning brains. Like, do you have any brains I could section? And you're like, sure. But then they find cool things. Like, the brains are even different. This is just the proportion of the total brain volume taken up by the medial cortex. And the medial cortex in lizards is analogous structure to our hippocampus, which is involved in spatial navigation and spatial memory. Like London taxi cab drivers have really big hippocampuses or hippocampi? I don't know. Okay, they have really big ones, London taxi drivers. And this corresponds to prediction that we made about how the spatial navigation abilities there might be pressure in a more spatially complex environment versus a more sparse environment. And so we don't know that all of these are evolved changes. Some of these certainly may be plastic. But the point here is that the white and the dark lizards are really, really quite different in basically every axis that we can measure, suggesting that even if they're not all evolved changes, that they certainly live in environments that have very different environmental pressures and selection pressures. So another part of the story is that we were also able fairly early on to identify one of the genes that's involved in color variation in this system. I am absolutely certain that it is not the only gene involved in color variation in this system, but it's a key player, and this is the melanocortin-1 receptor gene. And all 3 of the white sand species have mutations in the gene. And in 2 of the species, we were able to confirm with functional assays where we actually started with a wild type sequence and mutated just that site and then put it in a mammalian expression vector and did the whole thing. And we were able to confirm that mutation actually would shut down cyclic AMP synthesis, which is in the melanin. Okay, so we're confident that we know at least one of the major players in the melanin synthesis pathway at white sands. There's an interesting twist because in one of these species, this mutation is recessive, and in one of these species, this mutation is dominant. And that's actually kind of cool because our functional assays also showed that those would be the functional effects of those specific mutations. And it opens up a whole rabbit hole that population geneticists might care about and the rest of you wouldn't, which is the questions about the dominance of alleles that are involved in adaptation in the wild. Okay, so we're going to just So this is just the results of a recent project trying to test some of those ideas, not only about dominance and adaptation, but also just trying to get a better sense of how old are these adaptive alleles on this novel white sands formation, and what is the evidence at the genetic level for selection, right? So we have great evidence at the organismal level that this is a strong selection environment, but we didn't have, and we had an association between genotype and phenotype, but we didn't have evidence at the molecular level that selection had occurred in the genome. So we just finished a recent project. I'm not going to go through the details of this methodology, but basically we sequenced, we sequenced a big chunk around MCNR. So MCNR is a little gene. It's just like 1,000 base pairs long. But we wanted to see what patterns look like all around that gene. So we sequenced like 50 kb around that gene, and then like 50 kb chunks in lots of other random places in the genome. And so then what we get is this is just one way of looking at the data. These are FST plots. So FST, when FST is low, we're comparing dark lizards to light lizards. When FST is low, there's basically no genetic differences between the dark and the light lizards. If FST is high, basically the dark and the light lizards are quite distinct genetically at that place in the genome. And so what you can see is that FST in the same populations is very high for contigs that contain MCNR, but very low generally for other random regions in the genome. So what that means, if you are doing like an FST outlier test, MCNR lights up right away as like something different is going on at MCNR than is going on in the rest of the genome. And if you do more explicit tests for selection, you don't have to really worry about the y-axis here. It's a composite likelihood ratio, which doesn't really mean anything to your average biologist. But what we are looking for is when the composite likelihood ratio is high, that's evidence for selection. And these are kind of the position in the MCNR contig. So if you're walking along MCNR, this bar is where the coding region is, and then you're walking. So we're looking at whether there's selection that's observable at or near MCNR. So if you look at this in the two species, the whip-tailed lizards and the fence lizards, in the dark populations, there is no evidence for selection at MCNR, which we weren't expecting. So that's good. And then if you look at the light populations, you see these really, really high peaks in our confidence that there has been selection in that region of the genome. These are both highly significant, even though this one is lower than this one. And the interesting thing is that signature of selection is not localized just at the coding region, but the signature of selection extends quite a bit further upstream and downstream from the gene. And so that's something that people, in terms of thinking about genomic islands of selection, is something that people are interested in terms of understanding linkage disequilibrium drop-off and understanding what we can learn from the extent of this signature of selection. The other thing that we're trying to follow up on right now is there's also a little upstream peak here that we're interested in whether there might be any regulatory elements under there. Okay, so I've told you a little bit about this mutation at MCNR that is involved in color variation in this system. I've told you, even though I haven't told you in great detail, but we do understand some of the functional mechanisms of how this particular mutation could lead to color variation. There's this allelic dominance rabbit hole, and there's all this population genetics. But I really haven't told you anything about speciation at all. And in fact, it's something that we like to do is kind of, not we, and not like to necessarily, but there's kind of some little messy area in the Venn diagram between people that study adaptation and people that study speciation, and sometimes we're just not so clean about what we're talking about. Like, not all adaptation is the beginning of speciation, right? And certainly not all speciation requires adaptation. But I've always been interested in whether there's a possibility that the phenotypic divergence and the genetic divergence that we see at white sands could have anything to do with speciation, or is it just adaptation to different environments? Well, the reason I've always been curious about this is that melanin is a very important molecule in lizard life. It not only affects your dorsal color, which translates into how likely you are to be eaten, but it also affects the signaling color patches that are used for intraspecific communication and mate choice. So these are fence lizard bellies, but all three of the species have sexual signaling coloration that are influenced by melanin. And if you compare them statistically across populations, they're very different. And so I was always interested in whether MC1R could essentially provide a link between natural selection and sexual selection. In other words, if there's natural selection that has led to changes at MC1R, could those have a byproduct effect for who a lizard would select as a mate? And so this was a project that I did, well, I started many, many years ago. An undergrad field assistant said, do you think that the lizards know that they're white? And then that led to kind of like a whole summer of really weird experiments, most of which I will never look back on again. But this one was fun because we just asked the simple question, and then graduate students of mine have done this better over the years with better experimental design and in multiple species. Ask the question essentially whether there is mate preference in this system. Do the white sands lizards know that they're white? And so this should be, I think I have to click that for real. For those of you that are not familiar with lizard behavior, I just wanted to show you what we're scoring. We basically walk around, we find a male in its natural territory, we really quickly whip out the drift fence, put it around the male, but so he doesn't notice, because it's ideal not to bring the males back to the lab and then they're like, I don't even know who I am anymore, so well, I don't know. But they're just in their own territory, and then we introduce females to them in their territory and see what their behavior is. If you're not a herpetologist, you're going to get distracted by the fact that there's dental floss in this video. If you are a herpetologist, it's just not going to bother you. Okay, so this is our male. This is our female we introduced. The male's doing his little characteristic shutter bob, and he's approaching the female. Then this is a characteristic, they grab them at the nape of the neck before copulation. In this case, he's grabbing the dental floss, because there's dental floss involved in herpetology, mainly because we don't want to release this female into the wrong habitat, so we're keeping her on a tether. Okay, and then everybody gets released back to their own habitat at the GPS point we found them. They're all happy, and we have data, because we can go back and analyze these videos and measure the response of males to females of different color. And basically what you find this is just one way of showing the data, but in any behavioral metric, what you find is white sands males show greater courtship response to white sands females than to dark soil females. So white sands males prefer white sands females. An aside, just because I've been percolating about this kind of asymmetrical mate choice thing for those of you that might be interested, the dark soils males don't care. Which is interesting and not necessarily different than you would predict. If you're in a small isolated habitat patch, it might be more important to either have or have evolved preference in the smaller, more isolated population, where the swamping effects of gene flow might have a bigger potential effect. Okay, so basically, there's evidence certainly for rapid divergence at the phenotypic level and in some parts of the genome at white sands, but the different species don't exhibit similar levels throughout. So MC1R, everybody's really different, right? But if you look at other parts of their genome that aren't involved in the adaptation, the different species show very different patterns. They aren't all equally differentiated. So just with these three species, we've got the earless lizard that basically, even if you pick any random chunk of the genome, not MC1R, is quite distinct at any part of the genome, whereas the whiptail lizard is really fairly panmictic over this spatial scale. This depends a little bit on sampling and markers, but so one of the things that I'm really interested in this system now is moving into a more community scale approach to try to understand what are the factors that might accelerate or hinder divergence across species. What is it about the species biology? What is it about the way they experience that ecotone? What is it about their dispersal capabilities? And so we've been working on doing a community scale study of sampling phenotypes and genotypes of all the species that have these light adaptations at white sands across that spatial gradient. So we've just finished, and I should have included those, but we've just finished the crickets and the spiders, and moths and mice are ongoing. Nobody works at white sands. If anybody is interested in working at white sands, there's like nobody there. It's like I have to, I mean, I don't have to, but I mean, there's a couple other people, but it's like, I don't know the first thing about spiders, but anyway, they're cool. And the patterns so far are like radically different. So these guys are more like these guys. There's a total difference between the light and the dark populations. these guys, something's crazy going on. I don't know if there's cryptic species there, but they're all over the place. So I'm really excited about this community scale work. And I just thought I'd mention it just because of the grad student focus is it's fun to see how different people run their labs. And I would say that in any given year, I can kind of convince a small subset of the people in my lab to work on things that I'm thinking about. And then the rest of them are off making up their own projects, which is awesome. And so these are a couple of the other projects on this theme that grad students in my lab are working on. Madeline Gerard is working on these peacock spiders in Australia, which have an amazing, amazing divergence in color pattern and also divergence and convergence in color pattern across species. And then Karina Klonowski is working on these great mantellid frogs in Madagascar that also have similar questions about how color might play a role in these examples of rapid diversification. So the overall goal of kind of that half of the lab is really to understand how quickly adaptation occurs in novel environments and how predictably, in terms of the genetic mechanisms involved, adaptation and speciation might be across species. Great. So, everyone ready to be depressed? Oh, just a little depressed. Okay. So the flip side of what we work on is we work on some of the mechanisms of extinction that are really important in this day and age. Most of this focuses on wildlife diseases right now. It's not going to be a surprise to this audience that we're in a major extinction spasm right now. This is, as it should be, getting a lot of press right now. And there are, of course, many different factors contributing and interacting to threaten biodiversity on the planet. But I'm going to focus today on emerging infectious disease. This is a topic that I've been working on for some number of years. It's also nice to be in a place where I have friends working on these topics who know a lot more about fungal biology than I do, but I'm still my slot, unless you want to come up, Tim. Okay. So one of the things that's interesting about host pathogen dynamics in this day and age isn't just that there's host pathogen dynamics. It's also how these host pathogen dynamics might change because of other global change stressors, right? So as we're changing the planet, we might be changing these interactions. We're certainly changing the environment. We might, for example, if you take temperature, be changing a host pathogen interaction because we're changing the temperature that we're not changing necessarily the optimal temperature, but if we change the temperature, amphibians, for example, might be outside of their range for an optimal immune response, or we might be moving pathogens around, right? So there's a huge amount of interest in how global change stressors might affect species interactions. And this is just one graph, not from my group. This is a little bit outdated, but this was a study that looked at the number of emerging infectious disease events over time. And they tried to really control for reporting bias and other things that might artificially inflate this. But they found that even after controlling for things like reporting bias, that we are seeing an upsurge in emerging infectious diseases in human populations, in wildlife populations, and agriculturally important crops. And where I come in is that I am interested in amphibians as a study group. Amphibians are an incredibly diverse group on the planet. They have this colossal range in almost anything you can measure, from amphibians that are smaller than your fingernails to amphibians that are almost as large as me. They also have these incredible differences in life histories and reproduction styles. There's just a phenomenal diversity in amphibians, and some of you now know how to dance to the 7,000 Kinds of Amphibian songs, which I'm very happy about. But amphibians are one of the most threatened groups on the planet. They're certainly not the only threatened group on the planet, but as a group, as a whole, amphibian extinction rates are estimated to be about 100 times higher than historical amphibian extinction rates. And these are real, from a deep time perspective, real survivors that have survived the last few major mass extinctions on our planet. So these are mountain yellow-legged frogs. This is the system where I got interested in this work to begin with. I had colleagues that had been working on these mountain yellow-legged frogs in the Sierra Nevada for a number of years, and they started coming back and reporting that instead of seeing happy frog colonies like this, they were seeing really mass mortalities in their study sites, which for anyone who's been out hiking in the mountains, this is not typical, right? I mean, you don't find when animals die, they're not dying generally in these numbers, in these ways. And so this was being observed simultaneously all around the world. I wasn't working on amphibian declines at the time, but within a few short years of seeing mass die-offs occurring on multiple continents, this pathogen, Batrachochytrium dendrobatus, was described. And I think, oh no, I just got this letter from a kindergartner. Come on. Dear Dr. Bree, do you know about yellow-legged mountain frogs and other types of frogs? It's just like really cute. Okay, so I'm going to tell you what I know about mountain yellow-legged frogs and other types of frogs. This knowledge is relatively incomplete. There's an incredible amount that we still don't know about this system. Many of you I know are fungal biologists, so you don't need this, but I thought it would be nice just to show you how beautiful, oh, that shouldn't have sound, how beautiful BD is. This is the fungal pathogen we're talking about. I'll call it BD for the rest of the talk. These are these motile zoospores. They're aquatic. When they insist, they develop into these zoosporangia, where new zoospores are formed, and then they have a little release tube, and the zoospores are released, and they can infect amphibians. Chytrids are an ancient lineage on the planet, and most people just refer to BD as the chytrid that's killing frogs, but in fact, there's tons of chytrids around the world, and most of them make their living as decomposers in the environment. This is one of the only known species that has evolved a way to be a pathogen of vertebrates. And BD has now been described on every continent where there are amphibians. This is just a little heat map showing roughly the number of positive tests for BD in different parts of the world. And I got interested in this system because I had just started my postdoc at a time when genomics were not being applied yet to model organisms, which sounds kind of crazy, like, because now it just, but that was like, people weren't doing it. And so I was really looking for a place. I was making a transition from grad school to a postdoc. You can do this in any way, but my way was like, I want to develop a new skill set. And so I wanted to go into a bioinformatics lab and try to retrain. And at that time, the ways to do that were to kind of go into a fruit fly or a yeast genetics lab. Because those were the people that were really doing high throughput bioinformatics. And so I had this question of whether we could use genomics to understand more about the mechanism of this host pathogen interaction and try to answer some of these questions that were outstanding. For some reason, I saved this screenshot around that time, and I now look back and I think it's so funny. It's April 2003. Will the era of the genome begin in April? It was like so specific, and at the time I was like, I don't think so. Like, I'm not seeing any signs, right? Like, if you were working on non-model organisms, you know, in the late 1990s, this wasn't an everyday thing. It was like, yeah, sure, the era of the human genome has already started, but the era of the everyday genome hasn't really started. But certainly has since then, and so now we can routinely use genomic data sets to answer even time-sensitive questions. So I'll just give you a few vignettes about how genomics has helped inform our understanding of this BD system. It's certainly not the only approach that's been critical in this system, but it's what I happen to work on, so it's what I'm sharing. So this is a study that I actually did with Tim and Tommy, which is fun, and I think it might be the first time I've ever co-authored a paper with someone that I had never met until yesterday. So that was a real delight. And so this is a study that we did part of this is a study we did together a number of years ago, where we sequenced whole genomes of BD isolates from around the world. We also sequenced a whole genome of the closest known relative of BD. That's a chytrid that doesn't infect vertebrates. It's not a very close relative, but it's the closest known relative we had in culture. And this is just, we'll skip that. Okay, so before I show you the results of this, I just wanted to set up a little dummy data for you. For those of you that don't, there was this debate about whether BD is a novel pathogen, one that has evolved very recently and spread around the world in a flash, or whether BD was an endemic pathogen that's been lots of places, if not everywhere, for a very long time, and something else changed in the environment, or something else changed that essentially altered the host pathogen dynamic and made it more of a virulent thing. So this is just dummy data. Got a picture? Okay. This is a subset of all of the data, but it was what could fit on the slide. Okay, and you could imagine that if it was endemic, you might see some spatial structuring by geographic locality, or you might even see spatial structuring by host population. I want to vote. Okay, raise your hand if you are going to write this paper up and say that BD is and a rapidly emerging, just spread all around the world pathogen. Novel pathogen hypothesis, no support. Okay, raise your hand if you're going to write your paper and say that this is a endemic pathogens, kind of been everywhere forever. Okay, you guys are as confused as we were because we got these data back and we were like, what is going on? And so after thinking about this and doing many different analyses, and certainly subsequent data are shedding light on things, I think the important things to understand about what we do know about BD's history is that this tree doesn't conform really neatly to either clean set of expectations. So BD's evolutionary history is probably more complicated than we had been appreciating up to this point. We were treating BD as just like this single thing. And one of the things that we really were able to start thinking through more clearly was how there are certainly multiple lineages within BD. From the data we have now, there isn't a really clear point source of BD's origin or a really clear linear story about exactly how it spread. Certainly in some parts of the world there are. We know in some parts of the world we have really detailed epidemiological understanding of how it arrived and where it spread. But as a global presence, there are a lot of unanswered questions, including a lot of unsampled diversity out there, which I think that Tim James' lab is going to resolve for us, like lickety-split. So the other part of how genomics has been useful in studying BD is not only to try to reveal some things about the history of BD in terms of its evolutionary history and its invasion history, but also trying to start to understand something about its genome dynamics and whether there are parts of the genome that are doing interesting things. So remember that the hypothesis here is this is a relatively recently emerged pathogen in a clade where there aren't other, really, except now there's one other one, but aren't other vertebrate pathogens. So how is it doing this from a genome perspective? So I just want to show you a couple of graphs here. These are a little bit complicated, but what I'm showing you here, listed down here, are a bunch of different isolates that we've done genome sequencing for. And what I'm listing over here, actually I think you made the original version of this figure many years ago. We've added a bunch more isolates on there that you may not have seen yet, which are kind of fun. But these are supercontigs in the BD genome. You can think of them as chunks of chromosomes. And then the color coding is basically how many copies. So do you want to do another vote? Is BD a diploid? So this thing is a very strange critter, right? Because not only we have some isolates that are really consistently have 2 copies in each chromosomal segment. We have some isolates that are quite different from that and also even have differences within their own genome in terms of the copy number that you find different chromosomal segments on. This is my favorite. These are 3 new isolates that we just sequenced genomes from. You'll notice they all have the same name because they're all the same isolate. The only difference is how long they were kept in the lab. So here we have isolates that were just kept for a couple dozen generations in fungal terms, not a ton. And we see major differences just in the copy number of different chunks of their genome, right? So the interesting thing about this story is that in this passage history, virulence attenuated over time. So this old isolate could kill frogs, and this young isolate didn't. And so over a short period of time, you can have massive changes in virulence, and you can also have corresponding changes at the genome level. We don't yet know exactly what genomic changes might be linked to virulence, but we know that there's a lot of dynamic activity in the BD genome. We also know that part of this dynamic history, whether it's from the same processes of kind of big chromosomal segment duplication or not, is we see massive expansions in several gene families. So this is that chytrid that's a close relative of BD, and this is BD. So these 3 protease gene families are gene families that have been implicated in fungal pathogenesis in other fungal pathogens like ringworm and tinea in humans. And then this crinkler-like gene family, to my knowledge, is not even found in any other fungi. Now it is. Okay. Yeah, at the time it wasn't. So these are mostly found in oomycetes and, right? So we see these massive expansions in gene families that are thought to play a role in fungal virulence and other systems. And so one of the things that we've been doing, although I won't show you the data for it right now, is now, so these are from genome sequencing data, is now taking an expression approach where we're looking at the induction of these genes in different conditions, the induction in different environments, the induction in different life stages, and trying to get a better understanding of whether any of these genes might actually play a role in BD pathogenesis. Okay. So I'm going to really quickly just mention a few things from the flip side. We've also been doing a lot of work in the hosts so that we can try to understand how BD is affecting its hosts. I'm not going to show the genomics data. I'm just going to show kind of the results and how they map to what we know from other physiological studies and organismal studies. So the way BD is killing frogs is essentially by disrupting the physiological functions of the skin. So amphibians do a lot of things through their skin. They breathe, they osmoregulate, they electrolyte balance. And one of the things that we noticed early on in our genomic data sets is that we weren't seeing any induction of genes in immunogenetic pathways. We weren't seeing, if you've got a sick frog, we weren't seeing genes that should be involved in a robust immune response being turned on. We weren't seeing that at all. Since then, one of our colleagues has discovered that BD actually does secrete a compound that can kill amphibian immune cells. So there's some kind of really interesting interplay between this pathogen and the host immune system. So we were studying mostly susceptible frogs, and we kept finding this, that they're not mounting an immune response, they're not mounting an immune response. And then I had one of those days where you come to work and you're like, I think I might have been doing the wrong thing, like for the last 10 years. You know, you're like, I've been focusing on all these susceptible frog species because they're the ones that people care about and are asking about and they're of conservation concern. But like, we're not studying the frogs that are thought to be more resistant. So we don't have a comparison of like, what are the winners doing, not just the losers. And so that's what we've been embarking on the last couple years, is doing these paired experiments where we compare susceptible and resistant frog species that are reasonably close relatives to try to understand what they might be doing that's different from each other. And the short story is there's massive differences. If you take bullfrogs and you compare them to Bufo boreas, their trajectory once they're infected with BD is radically different. I think I'm going to flip through the details of that in the interest of, I think you guys wanted a few minutes before 5 for questions. So I'll skip the details of that, but I'd be happy to talk about that later. These are mostly lab infection experiments. We can look at BD loads over time, and we can really confirm in controlled settings that some frog species are more susceptible than others to BD infection. We can measure things like whether or not they lose body weight, what the morphology of the skin is, what genes are turned on and off. And one of the things I'm most excited about right now is going back to some of these species that we had classified as highly susceptible. And we are now finding 10 or 15 years later, after those enormous population crashes, that some of the populations are recovering. And so that's leading to kind of the last piece of this puzzle in my lab for right now, is trying to understand whether there could be evolved changes either in the host and or the pathogen that could help explain some of these recoveries. We don't need evolution to explain these recoveries. There could be demographic effects or other environmental effects. But what we're doing right now is we're comparing historical samples of BD and frogs to contemporary samples and trying to understand whether there have been any interesting changes in their genome that correspond with increased resistance from the frogs or decreased virulence for the pathogen. Blah, blah, blah. We're also doing a little bit of white-nosed bat stuff. But the hope in terms of conservation implications is that this is a figure that we had put together just for a little review paper, is that if you think about, and you could do this for most any system of conservation interest, particularly when it's disease-related, that in the BD system, between the time that BD was first, that frog die-offs were first observed, and then BD was first described, and then there were real action plans in place, it was a pretty long time. And one of the interesting kind of meta outcomes of having a community that's more mobilized around emerging infectious diseases of wildlife is that it's not that all of us started working on white-nosed bat, but there was like a scientific community ready to support each other in a way when the first bat mortalities were observed relative to when the first frog mortalities were observed. And that time period, I mean, this might be a simpler system, there could be some other explanations, but the time period between when the first bat mortalities were observed and when there was real on-the-ground conservation action was much shorter. And so that, I think, is at least a small encouraging thing about how our science can eventually have a conservation impact. Okay, so I'm not going to be exactly 4:55, but I'm going to do my best to just zip through these last few slides. So here we are. And whether we're thinking about the potential for adaptation or the likelihood of extinction for a proportion of biodiversity on our planet is undergoing massive changes. Land use, urbanization, consumption. This is an awesome photograph. Has you guys done the YouTube thing where you watch the planes take off? And you should do that if you haven't. But these are globalization. These are just the pathways for transit on our planet. It's like covering most of the planet. And I teach global change biology as my core undergrad class, and I've noticed this ethos in the generation of undergrads that has grown up understanding that we're at kind of a scientific and planetary moment of crisis, that there's this real attitude of like, oops, we suck. Like, humans suck. And as like we have evolved, we have basically like shrunk the capacity for healthy ecosystems. And I just love this because it exemplifies it exemplifies actually the worldview that most of the undergrads I come into contact with come into my class with. And so we talk a lot about worldviews in that class. We talk a lot about how do you take the scientific knowledge we're generating and try to understand what it means about our place on the planet. And we talk about kind of the, oh, we're awesome worldview versus the like, oh, we totally screwed everything up worldview. And one of the things that I have really enjoyed in reading some of the kind of ecological philosophy literature is the emergence of, and I'm sure there are others in the room, but the emergence of at least a third worldview. And I'm just going to use one example. This is Joanna Macy. Have people heard of Joanna Macy? Yeah, so this is just a quote from Joanna Macy, who compares this great unraveling, the like we screwed everything up idea, with this thing that she calls the great turning. And this is a quote of hers that I'll just read to you. The most remarkable feature of this historical moment on Earth is not that we're on the way to destroying the world. We've actually been on the way for that for quite a while. It's that we're beginning to wake up, as from a millennial-long sleep, to a whole new relationship to our world, to ourselves, and to each other. Now, that might sound a little New Agey to you, but then I was just rereading Darwin's grandeur quote, which is well known to many people. There's grandeur in this view of life, endless forms, right? And I was struck by the guy sitting next to me on the airplane who was looking over my shoulder and was like, who had no idea who Darwin was. And he was like, whoa, is that guy like a yoga teacher or something? Because that sounds like way out there. And I was like, wow, that's really, it's kind of awesome and fascinating to like look at Darwin in that way. But it just kind of struck me in terms of this life being interested in life mentality, that we're all coming into our professional lives, especially you guys, because I'm mostly here for the grad students, is like, especially you're starting your career. Like, you don't have to start it with the like, everything sucks and we totally screwed up mentality. There are other possibilities going back to our roots in our field, but also going back to what I think we know to be true as a shared community of humans, which is that there's some responsibility and some agency to communicate what we know and to be involved in making a better world to some extent, in some way, in whatever way happens to find you in your life. So with that, I'd like to thank my lab members that contributed to this talk, a couple of key collaborators, and then if we have just a couple minutes, I'm happy to take questions and looking forward to more time with you all later today and tomorrow. Thanks.
So we definitely have time for questions for Bree.
Yeah. Yours isn't going to be as hard as Tim, so I'm going to go to you first.
So this question attempts to integrate both parts of your talk, but with your MC1R story, MC1R has been a target repeatedly for mammals and birds in pigmentation evolution. And I guess the first part of my question is, for the adaptation to the white sands story, have there ever been any like reversions back to like a more darker phenotype? And as part of that question, and this idea of adaptation being predictable at the level of the genome, do you think we'll ever get to a point where we can use genomics to help us understand certain species that are at more risk to extinction, given their capacity to adapt to new environments?
So the first half of the question is easy, not that I know of. We don't have any kind of back And I think actually it would be a little hard with the resolution of the data we have now to unambiguously say that that's not migration, like they're not alleles kind of filtering in from the dark soils populations rather than reversion. I mean, you could tell that if you had enough genetic data, but we've never seen that or know of it. I think the other question is a really interesting and really hard question. A number of years ago at the amphibian declines meeting, I remember kind of one of the leaders of our group saying, you know, if we gave you a lot of money, could you use the genetic work you're doing to generate a predictive map for us about, like, so we don't want to have to collect BD specimens and wait and see what's going to happen with the frogs. We want to send the BD specimens to you, have you genotype them, and then tell us if we have to worry. And it was kind of like, no, I can't do that. Like, for me, and other people may be working in systems where it makes sense, but for me, the genomics work is never going to replace the on the ground really, really understanding these critters and understanding the biology of the organisms and understanding the context, both the biological context and the cultural context, and, you know, everything that's happening, especially if you're trying to manage for an emerging infectious disease. It's actually not only about the biology, it's also about the political and cultural context where that's unfolding. So I think that's a rich conversation and one that would be fun to continue with all of you. But in my work, I don't feel like it is, nor am I really looking for it to be that replacement. That doesn't mean that we're not interested in, like, finding the predictable elements and finding, like, what are the well-trod pathways of evolution. Yeah.
My question was actually about MCs, whether the mutations that you saw were circulating and sort of pigmented form or low.
Yeah. So we've done all these simulations that suggest, especially for the recessive form, that it basically has to have come from standing genetic variation in terms of kind of, you know, being at enough frequency, intermediate enough frequency that it could become visible to selection in the time frame. Like, we've done a lot of simulations that suggest that, but we have never found it circulating at low frequencies in heterozygote form in the dark soils populations, and our estimates of the age of the alleles from not just the Mcir data, but the longer chunks are actually not that old. So I'm rethinking that a little bit. They're not 2,000 years, like the estimates, and there's confidence limits obviously around that, but the estimates are more like 7,000 years or something. So so that's an open question for me, the kind of adaptation from standing genetic variation versus new mutation. There's very clear theoretical predictions, and when we do simulations with this system specifically, the simulations conform to the theoretical predictions, but then the real data are a little more nuanced than that to me. Yeah. Yeah.
I was wondering if you had a hypothesis as to why even in one strain of EEB you see this change in copy number. Is it just some crazy transposable element that's running around the genome?
So a mechanism in terms of, like, the actual genomic mechanism for copy number changes, or more like a possible reason? Like, if you're, I mean, so I think the convenient thing that people talk about, although we don't really know, is that if BD is cultured in a lab environment rather than on a host, there's a possibility that selection is relaxed in some way such that, you know, you're not weeding out these crazy things that happen. But I don't have any, do we as a community have a deeper understanding about how, yeah, I mean, the actual mechanism of why that's happening? I don't know. Stay tuned. We're going to write a paper about all the things we don't know. It's going to be, like, published in the Journal of Irreproducible or whatever research. Great. Thanks so much for coming out.